Hello, my name is Ellen Berg, and I'd like to welcome you to today's webinar. I'm the CSO of BioSeq, a division of DiscoverX, and today I'll be telling you about the application of the BioMAP platform, which is a phenotypic screening platform for characterization of drug safety mechanisms in human primary cell disease models. Now, researchers in drug safety and toxicology are well aware of the challenges of predicting human drug toxicities. The mechanisms are diverse. Most remain unknown. There are few targets of toxicity that have been identified. Animal studies are limiting. Not only are they costly and can take a long time, but there are species differences such that the data that we get in animals is often not predictive of humans. Certainly, clinical failure rates have not been improving. Approximately 20 to 30% of drug candidates fail in clinical testing, and here is shown some failure rates for different reasons in phase 2. Certainly, we need better tools, ideally tools that would have better human physiologically relevance and that would help us understand and determine mechanisms of toxicity. So the platform I'm going to tell you about today, the BioMAP platform, is an in vitro testing platform in primary human cell-based tissue and disease models. Our approach has been really an evidence-based approach, unbiased, data-driven methodology. And what I mean by that is we are letting information that we've gained from the clinic or from animal testing on large numbers of drugs and test agents reveal to us what's important in predicting human toxicities. Applications are diverse for compound and target validation, translational research, and drug and toxicity mechanisms of action. The platform consists of three pillars. There are the assay systems themselves that were developed using exclusively primary human cells in single and co-cultures. We've developed over now 50 model systems. I'm going to tell you today primarily about our flagship panel of assays, flagship panel of 12 assays, the Diversity Plus panel, where we've developed the largest reference database of profiles in that panel, and our predictive informatics tools that we've built to provide you with mechanism and knowledge about agents that we profile. The key features of the assay systems, as I mentioned, primary human cell types. Why primary cells? These are the closest you can get in vitro to the human tissue. We put these cells into physiologically relevant context, and what I mean by that is that we put them in complex activation settings where we add in the factors to mimic the tissue or disease biology, to better mimic how a patient is going to respond. Many of our assays are co-cultures. They have multiple cell types because we know in tissues, cells talk to each other. And so this makes a better model system to capture those interactions. The final point about these assay systems is the endpoints that we're measuring in these assays are each themselves already known clinical biomarkers. So they already have a wealth of information about how they behave, respond in patients connected to these endpoints. They are primarily protein biomarker endpoints. Now, some of the reasons why developing a human-based system is really important is we keep in mind how different animals are than man. Many of our safety assays, in vivo assays, are in the mouse or in the rat, are in non-human organisms. And if we compare ourselves to a mouse, we live a lot longer, we weigh a lot more, and we are exposed to a very dirty environment. some of the key processes that are very different between rodents and man include DNA repair mechanisms, because of course somebody who's going to live 70 years and be exposed to sunlight and other damaging agents, we have a lot more repair mechanisms than mice do. And so these mechanisms are quite different. We are substantially larger than mice, and our mechanisms to control blood flow and hemostasis are a lot more complex than mice have and rats. And our immune system status is very different as well. Most of the immune cells that are traveling around our bodies are activated in memory cells because we are constantly exposed to all kinds of insults from the environment, viruses, bacteria, dogs, children, etc. So these make this very different. So when we put these assays together, this is how we think about putting together the biology. We start, as I mentioned, with human primary cells. We put them into co-culture with cocktails of different proteins and growth factors and substances to result in a particular cell system. And then what we do is we test this cell system by measuring clinical biomarkers. Do we have the right appropriate cell types and the activating conditions to induce expression of clinical biomarkers we already know are expressed in the disease or in the tissue setting and are important? And we continually not only look at one biomarker, but we look at a panel of biomarkers. And then what we do is we iterate the process. We change the cell types, we change and look at the panel of responses, and we change the activators and optimizing the system so it is the best model of a particular disease or tissue state as we can manage. And then furthermore, what we do is we then validate the system with, for example, well-characterized standards of care, those agents for which, or compounds or drugs for which we have clinical data and can confirm whether or not these assay systems are behaving as expected that the tissue does in the patient. And at that point, we have a system that does a pretty good job of modeling a particular aspect of a tissue or disease setting. And we don't stop there. With just one system, we have actually developed panels of systems that each model a particular aspect of human tissue or disease biology. And we've put together a collection of 12 of these systems in our Diversity Plus panel that really covers a broad range of human biology and really in activation conditions that model adaptive responses to the environment, whether it's of stress or bacteria, et cetera. And so we have assay systems that contain vascular cell types, including endothelial cells, smooth muscle cells, dermal fibroblasts, we have epithelial cells, keratinocytes, and then we cover quite a lot of immune biology in this particular panel. We have peripheral blood mononuclear cells and assays that capture T-cell responses, B-cell responses, monocyte and macrophage responses. So it covers a broad range of tissue biology that will be important for capturing the kinds of responses that we make to drugs when we take drugs or to the environment. So I'm going to walk you through what happens when we test a compound through this panel of assays. How do we look at a profile, and how do we interpret the data, both on individual endpoints that we might measure, and then how we analyze the entire profile as a unit. And here I'm showing you the BioMAP profile of an approved drug, Trametinib, which has been approved for the treatment of metastatic melanoma. And you can see across the top for the BioMAP assay systems. Along the bottom are the protein biomarker endpoints that we're measuring the levels of. In every assay system, we do measure whether or not the drug or whatever we're adding to the system is killing the cells, because when you kill a cell, you will change protein levels, and that will have a very different meaning than if the cells are healthy and we're changing the protein levels. So we do measure overt cell cytotoxicity in each one of these systems. The reason is there are many different mechanisms of cell toxicity, and different cell types in different environments will be sensitive or susceptible to those different mechanisms. We then report the data as a log expression ratio. We measure the level of each of these proteins in the presence and in the absence of the drug. We take a log ratio, and so if the level of the protein is increased in the presence of trametinib here, you'll see a peak above the dotted line, and if it's decreased, you'll see peaks below the dotted line. And the gray zone that's surrounding the origin there is showing you the 95% confidence envelope, which is the 95% of negative controls are within that gray zone. And so any point that's outside of that zone has a 5% chance of being false positive. So the four colors, as you see here, red, orange, yellow, and green, represent different concentrations of trametinib. And you can see, while there's a dose response, there's a concentration response pattern to the profile here, that the overall shape of the profile is consistent over multiple concentrations. And that's a feature, this pattern, that we'll talk about a little bit later. But just to give you a heads up that other compounds that are also MEK inhibitors will give you the same shape profile. So what's really interesting then, of course, is what are all these peaks and valleys? And remember, I mentioned that these are all clinically relevant biomarkers. Every single activity here tells you something about how this drug is behaving in the clinic. So let's walk through that. The first thing we see is activities that are relevant to cell cycle, so inhibition of endothelial cell proliferation, T cell, smooth muscle cell, and fibroblast proliferation. These are activities that are, we know that are controlled by and regulated by the target of trametinib, MEK kinase, and we see them here in the BioMAP profile. We also see effects on markers that play a role in angiogenesis. We know that trametinib inhibits angiogenesis, that's the growth of blood vessels in vivo, and so we see that recapitulated here. We see a number of changes in protein markers that are in proteins that are involved in tissue remodeling, which include MMPs, these are matrix metalloproteases, plasminogen activators, and all of these three categories of activities are relevant and important and involved in trametinib's ability to be an effective oncology drug. So these are all activities that are related to efficacy of trametinib. But we also see a number of other activities. We see inhibition of a number of acute pro-inflammatory biomarkers, IL-1, TNF, and IL-8, as well as inhibition of T cell activities. These are all proteins and biomarkers that are involved in fighting off infection. And we do know that patients who are taking trametinib are at increased risk of infection. And so we also see the activities in the profile. We see, in addition, upregulation of a protein called serum amyloid A. This is a protein that's a risk factor for incidence of cardiovascular events. We know that trametinib is associated with potential for cardiotoxicity. Most oncology drugs are. It's not a surprise. It needs to be managed, but we see the activity here. And then we finally, we also see an activity in our fibroblast-based model of wound healing. We see increased levels of VCAM, vascular cell adhesion molecule. This is an adhesion protein that's involved in the recruitment of chronic inflammatory cells into sites of inflammation. And we see increased levels of this protein, and we have associated that increase with a risk of skin rash. The patients who are taking Trametinib have one of their major side effects is a skin rash, and clinicians actually use that evidence to support the efficacy of the drug in patients. And of course, if you're fighting from cancer, you're willing to tolerate a little bit of skin rash. And so in this way, we see activities related across the panel here, important for efficacy, and as well as related to safety issues or safety features related to this clinical drug. And so, we can see here the correlation of these activities with clinical effects. Keep in mind the power behind this platform is the fact that we have a very large database. We have more than 4,000 agents that have been tested and profiled through this platform, drugs, clinical stage, approved and failed, experimental chemicals, biologics, and even materials. Materials are active, nanomaterials. We can test mixtures, extracts, etc. And you can imagine that once having a very large database with all of these materials and compounds profiled, you can start to really build knowledge about biological mechanisms. We've done mechanism prediction, which I will talk about, and clinical and preclinical correlations, particularly with safety, which this presentation will focus on. And keep in mind that this platform, particularly our Diversity Plus panel, which is a broad standard panel of assay systems, is very, very useful for connecting projects across therapeutic areas. And often we start a project in one therapeutic area, and then we'll realize that it could be useful in a different area, and then we have to transfer all the knowledge from one therapeutic unit to the next. This platform makes this very easy to do and very easy to find other indications where a drug might be useful. So, as keep in mind, I mentioned about using the patterns of these activity profiles to make new knowledge. So one of the patterns that I want to talk about now is the fact that we can capture concentration response patterns. And so here's an example of a very sharp dose response pattern. At every concentration, you get a different shape profile, and between the red and the green, you go from completely almost inactive to very strongly active in every system that's shown here. And the arrows, the black arrows that I'm showing here indicate that at the highest concentration, this compound is actually cytotoxic in every system. So that's a type of a concentration response pattern we might see. We'll see inactive profiles. We'll see highly selective profiles like this one, and we will see profiles that look like the one in the bottom right corner. Similar to the Trametinib, we see the same shape profile of this compound across multiple concentrations. This is a feature that we call being dose-resistant or concentration-resistant. It is a feature of late-stage, highly target-selective compounds and drugs, particularly safe drugs. And what we have been able to do is to take this concentration response pattern and start to look at how it tells us about clinical exposures. So on the left here is a list of a few known drugs and their mechanisms and their maximal clinical exposures. That's the human data on the left. On the right is our BioMAP in vitro data. What I'm showing in green are the concentration ranges for each one of these drugs at which the compound gives an active profile in BioMAP systems in the Diversity Plus panel and non-cytotoxic. And you can see that there are some compounds, some drugs like fluticasone, rapamycin, and trametinib that have a very large concentration range at which they're active and non-cytotoxic. And other drugs, such as cisplatin, sorafenib, and sunitinib, who have a much shorter range of activity in our systems. We see that, as I'm plotting here, these are the concentrations at which some of these agents become cytotoxic. Some compounds we have not reached cytotoxicity or have not tested beyond at higher concentration. And we can start to then build a ratio of therapeutic window. Then what we can do then is overlay on top of that what we know about the clinical data. So these are the average, they're in red, are the average clinical Cmax values for each of these drugs. And you can see for some drugs, they've got a very broad range of concentration at which they're safe and active, whereas things like cisplatin and sorafenib and sunitinib, the difference between the concentration at which they're exposed in the clinic and their concentration at which they become cytotoxic to cells in our systems is very narrow. And we have been able to correlate this with a safety window. And we can use this feature for prioritizing drugs. And we know, for example, that among the kinase inhibitors, sorafenib has more toxicity than sunitinib, and sunitinib has more toxicity than trametinib. And so we can actually watch the progression of classes of molecules for which we have then, our pharmaceutical and biotech clients have developed drugs that have, better safety profiles and improved therapeutic windows. And what I'm telling you here is that this platform allows you to pick this feature up in an in vitro setting in a system where we can run hundreds and hundreds of compounds and don't have to run a lot of animal tests to be able to identify and select and triage compounds that will have a better safety window in vivo. So going back to patterns, of course, there's another type of pattern that we look at, and that is the pattern of activities across the profile. And we can look at profile similarity as and its relationship to information such as mechanism of action. If compound profiles are highly correlated, they'll have a high similarity correlation, a metric, in this case, a Pearson correlation. And if they're not very correlated, we can, mathematically identify that. And we can compare, because we have a large database, we've run a lot of compounds, we can do a very large comparison, pairwise comparison of a number of different compounds and drugs. And when we've done this, we've been able to show that, compounds with the same target mechanisms will give the same shape profiles and tend to cluster in this type of cluster graph. And here I'm just showing you some of the example profiles, and you can appreciate, how very different some of these mechanisms are from each other based on the activities, the systems, and the activities that are characteristic. And moreover, not only do compounds with the same molecular target mechanism tend to give the same shape profile and cluster together here, but we can also pick up pathway relationships, because maybe we won't have a representative compound with a particular target in our database, but that target likely exists in a pathway, and we will find the most similar, or the pathway to which that compound belongs. So that's a very useful application of the platform to evaluate profiles for their shape and to identify potential mechanisms of action just through similarity. We've taken this work beyond just a similarity search, and we've taken advantage of the fact that for many mechanisms and targets, we have multiple compounds that are selective for that target that we'll have run and be able to compare those profiles and actually look to see if we can build a consensus profile for that mechanism class. And for many mechanisms, we've been able to do this. We can actually, convert this into a HeatMAP form. This allows us to compare a lot of different mechanism classes to each other, and we can use those consensus profile signatures as to reflect, what is the target-specific biology. So we have developed this mechanism HeatMAP, which is a new deliverable for our Diversity Plus panel service. Along the right side are each of the individual system and all the different biomarkers that we're measuring in each assay system. Along the bottom are the mechanism classes, so including the MEK inhibitor, or the P38 inhibitor class that I just showed you. So what we see is that vertical signatures then define the target mechanism. So here, this particular class here circled in orange is MEK kinase inhibitors. And then we can look at the horizontal signature. Here is how VCAM is in our fibroblast model system of wound healing is regulated by all of these pathway mechanisms. So it's a very, very rich plot, and one can do a deep dive in inspecting the test compound activities and how it compares to all of these pathway mechanisms that are shown here. A lot of biology in a very efficient format. So even beyond developing these consensus signatures, we've actually taken machine learning to build algorithms that will be able to predict give us a value, a number, classification, whether or not a particular compound it can be classified into one or more of these mechanism classes. The method is we generate, a test data set using compounds, multiple compounds and multiple profiles at multiple concentrations for each of these mechanism classes. We then build classifiers and then test the performance. We published this a couple of years ago, and we've demonstrated the performance of here, shown at the bottom of a few of these mechanism classes, and can now this mechanism classification is now included in our new Toxicity alert service, which is an add-on to our Diversity Plus profile service. Number of classifiers, here's MEK, includes all of these mechanisms here. These are mechanisms for which we've built classifiers. The ones in red might be particularly interesting from a safety perspective. There are known safety correlations with each of these mechanisms. Calcineurin is immunosuppressant, microtubule disruptor, mitochondrial inhibitors, et cetera, and even many of the other ones also have known either efficacy or safety issues associated with them. And do keep in mind that each one of these can, depending on the clinical indication, can be an efficacy mechanism or a safety mechanism. Cancer drugs, microtubule disruptors are certainly good cancer drugs, so we have to keep that in mind. So I hope I've shown you how this platform is very well suited for safety and tox applications. It covers a lot of biology and mechanisms. It's a standardized platform. It has a large reference database. The data is reproducible. It reproduces enough so that you can actually build mechanism classifiers that perform very well. Each of the activities, as I've shown you, correlates with very specific clinical effects, and in fact has been quite useful for us in discovering toxicity mechanisms. And that's what I'm going to talk to you about next. I'm going to walk you through a case study here where we have used the database to identify signatures that are correlated with specific human adverse effects, and the case study is a study for skin irritation, skin rash. So where we started with this program is one of our collaborators had some compounds in a CNS program. The target was a GPCR, and the program had been discontinued because there was a skin tox in non-human primates. And I'm sure you can imagine the cost of non-human primate studies. So there was unknown mechanism of toxicity, and they had actually tested two different compounds, at least two different compounds in the non-human primates. They both had the toxicity. One seemed to be a little more active than the other one, and their goal was, can we determine the mechanism of toxicity, and can we figure out whether or not it's on or off target? They did not think it was on target, but could we provide information about this? So what was really interesting is that when we ran the lead compound through our Diversity Plus platform, it was quite active. It was a bit surprising, a CNS target. And there were diverse, interesting activities, mostly in inflammation, immunomodulatory activities for this compound. When we compared the compound 47 and 88, we saw that there were in this system over on the left, there were a couple of shared activities, so this kind of pointed us in the direction of looking into this particular system as potentially being related to the common side effect. And what we know about this particular system is unexpected. It is a human primary cell co-culture. It has peripheral blood mononuclear cells and vascular endothelial cells. The system is stimulated with a low concentration of TLR4 ligands, so these are bacterial stimuli. So the biology modeled in this system is monocyte and monocyte activation, vascular inflammation, and responses to infection. And the key endpoints here that were shared by these two compounds include increased PGE2 and decreased TNF-alpha. So it turns out that this particular signature is common among some known irritants. It is shared by chemical vesicants, which are blister agents, retinoids, and also some VDR agonists. It turns out that retinoids and VDR agonists are known skin irritants, and in fact, dermal prostaglandin E2 is a biomarker for skin irritation, and chemical vesicant damage has been shown to be cyclooxygenase dependent. Now, cyclooxygenase is the enzyme that is the rate-limiting enzyme for prostaglandin production. And so we saw this signature with their compounds, and it is a signature that is associated with at least these known irritants. So we're able then to put all of our information together, not only with the profile from the client compound, but also the reference agents, as well as having profiled some of these mediators into a fully fleshed out model of how we think irritants, particular types of irritants, can cause skin irritation. And so here, irritants lead to cause increased levels of PGE2. PGE2 can change collagen ratios, PGE2 also promotes vascular permeability, production of IL-6, IL-17. IL-17 leads to increased IL-8 and tissue plasminogen activator. The changes in collagens and tissue plasminogen activator lead to a compromised barrier function problem. And the IL-17 together with IL-6 and IL-8 lead to and promote neutrophilia, so the recruitment of neutrophils into the site. And some irritants, including both retinoids and the compounds, the client compounds here, also increase eotaxin-3 in one of our systems. Eotaxin is involved in the recruitment of allergic type inflammatory cells into sites of inflammation. And so here we can put together this model of how we think the irritants and this particular compound are acting to cause skin irritation. So you get a much fuller picture of the whole biological mechanisms that have caused irritants to cause tissue damage in this case. So remember, not only can we get that nice picture, but we also have this very large reference database, which allows us to now test, do a more critical test of the association of this signature with the irritation response. So we can take the signature, run it through the database of all of our compounds and see, well, what other agents and materials have that particular signature? How specific is that for skin irritation? It turns out it's quite uncommon. Very small percentage of agents in the database have the signature. Interestingly, most of the agents or drugs that actually have human clinical data reported did have skin side effects. And it turns out that we were able to look at that list and identify specific common mechanisms that are associated with this signature. Not only retinoid and VDR agonist mechanisms, but also modulation of aryl hydrocarbon receptor, PKC activation, and thromboxane A2 synthetase inhibition. So we presented this recently. I'm not going to go through this slide in detail, but if you'd like to inspect all of the agents and the concentrations that we saw these activities, this is here for you to peruse later. But just to remind you that a very specific signature that was associated with skin irritation and was shown to involve one of five very specific mechanisms. So it's a mechanism-based signature that is associated with a clinical side effect. So what about compound 47? It turns out that our collaborator tested these compounds in nuclear hormone receptor assays and did find that notable induction of retinoid acid receptor responses and some changes in aryl hydrocarbon receptor. And so in this way confirmed, was able to take our list of potential mechanisms and confirm their compound mechanism. So not only do we have an in vitro assay and a signature for skin irritation, it's associated with 5 very specific mechanisms. We've used this information to detail the tissue mechanism at which that is responsible. And, for the customer, for the client, it allows them then to go back and find backup compounds that might not have this side effect for next generation compounds as well. And even if you have something that's already in the clinic, knowledge of these mechanisms allows you to design mitigation strategies. And for many toxicities, it's a feature of drugs that have to be managed and that can be managed. So for the skin irritation, there's the adverse effect, There's a set of reference drugs that define the signature. We have key biomarkers that are involved in the signature and a set of biomarkers that define the motif that we use and in a particular system. So not only have we put together, we identified this signature for skin irritation that I just went over with you, but we also have developed signatures for acute toxicity, for immunosuppression, for a particular type of liver toxicity, risk of organ toxicity, the MAP kinase pathway-related skin rash, skin sensitization, and risk of thrombosis-related side effects. And remember, not only do we have the signature and drugs that are associated with that signature, but we've also identified, particular pathways and targets, mechanistic pathways that we've identified for each one of these signatures. So these are mechanism-based tox alerts. So, in conclusion, I hope I've shown you that the BioMAP platform is a reproducible, standardized platform that has applications across a number of domains. It is useful for assessment of all kinds of materials, combinations, drugs, chemicals, and extracts. We have a number of examples. I've shared with you one today on mechanism of toxicity, but we've used this platform and these tools for lead prioritization, program rescue, and mitigation strategies. Just to remember that DiscoverX is a leader in cell-based and target-based assays. Our platforms cover GPCRs, kinases, interleukins, pathway assays, epigenetics, checkpoint, in addition to the BioMAP platform, which provides a phenotypic screen across all of these domains. I'd like to acknowledge the people on the BioSeq team, as well as collaborators from Pfizer, from the EPA, from the NIEHS, and Eli Lilly. And I'll close and take questions now. All right. Okay. First question is, what kind of classification method did we use? So the classification method that we used for the mechanism models was support vector machine. At the time, we tried a variety of classification algorithms and methods, trees, and a few other random forest, and a few other methods. It turns out that these mechanism classes are quite well separated, so probably a lot of tools might work and could work for this application. So I don't think there's particularly anything special about support vector machine, but it turned out that it worked in this case. Okay, second question. Are you working on additional mechanisms? Okay, so actually we're working on additional mechanisms for both our mechanistic classifiers as well as toxicity mechanisms. So the mechanistic classifiers, we depend on having access and being able to profile multiple compounds that might be selective for a particular target. And so we are continually looking for those and expanding the number of mechanisms that we cover. In terms of the tox alerts or toxicities that we're looking at, we are actively developing signatures in a couple of different areas. Cardiotoxicity is one area that we're working on and gathering information from our clients and interested academic collaborators. on, compound drugs that cause different types of cardiotoxicity. We are mostly we've not really focused on arrhythmias, but actually other types of cardiotoxicities. And we're actively interested in people's contributions to that project, so keep that in mind. And another area is renal tox. So we're gathering information and compounds in that area as well. So, and we do depend quite heavily on outside collaborators for, sometimes compounds, but really their knowledge of drugs that cause these particular side effects and the details of the exposures at which we see the side effects, the incidence, and the type of pathology or the type of toxicity that's observed. It's very difficult in people because you can't do a necropsy on people and get a full histopathology on patients sometimes because they do recover from many of these toxicities, and so we don't really know at the actual fine phenotype of the tox in some of these areas. Okay, a question on how do we handle genetic differences or genetics. So this platform we've developed for screening. So it includes, in our assays in the Diversity Plus panel, they contain cells that are from pooled donors. And so we are averaging the response, taking an average of a response. And what we are looking for are those mechanisms that would be important to many people and be population-based. The other way to spin this platform, which we've done in custom projects, would be to look at individuals separately, and that's a very interesting project to do, and we've done some of that work. One can even, start to look at genetic chain differences between donors and that sort of work too. The caveat is that they're quite variable, and it takes a lot of donors to capture all of the range of human responses, especially if you start to look at patient cells. So these are cells from diseased patients. Then typically patients are at different stages of disease, and they have been on different drugs. So it gets to be quite a complicated picture, deconvoluting patient-specific differences from drug differences in the patients that have been on different drugs. Another problem with all human cell-based assays is the fact that, and from patient cells, is the medical information that you can get on those patients is very limited and typically poor quality. And it's poor quality because we're people and we're not good at, always filling in all the boxes or coming into the clinic. When we said we would, we drop in and drop out. There's missing data and those sorts of things. So it makes building assays with patient cells, quite challenging. But by focusing on normal cells from normal people, where we are inducing the disease phenotype ourselves, we actually make this platform much more reproducible and much more broadly useful for doing these types of predictions. And I think that's all the time we have for today. I want to thank you for your attention. Please feel free to send us any additional follow-up questions. We'd be happy to take, and thank you again.